I am really happy to be hosting Noelle Antao, Joseph Sall, Gira Bhabha, and Alice Liang to discuss the recent PLOS ONE Lab Protocol paper entitled Sample Preparation and Data Collection for Serial Block Phase Scanning Electron Microscopy of Mammalian Cell Monolayers, and the associated detailed protocol in protocols.io. Dr. Gira Bhabha is an associate professor at Johns Hopkins University. She began her independent career in 2017. The Bhabha Lab works closely with the lab of Damien Eckert since their inception. The two labs have functioned synergistically as a single group. The Bhabha-Eckert labs study structural mechanisms of cell biology of microbes and their interactions with hosts, using integrative approaches including X-ray crystallography, cryo-electron microscopy, cryo-electron tomography, optical microscopy, biochemistry, microbiology, and cell biology techniques. Dr. Alice Liang is the director of the microscopy laboratory and a professor of cell biology at New York University Grossman School of Medicine. She currently serves as a council member of and biology director for the Microscopy Society of America. As the inaugural director of the Electron Microscopy Corps since its establishment in 2005, Alice has overseen its evolution from a general electron microscopy facility to a comprehensive imaging center. The Corps now integrates optical microscopy, volume microscopy, and correlative microscopy to meet the advanced imaging needs of NYU laboratories, as well as external academic institutions and industry partners. Noelle Antao did her PhD in cell biology at Weill Cornell Graduate School of Medical Sciences in New York City and is currently a postdoctoral fellow at the Bhabha-Eckert Lab at Johns Hopkins University. Joseph Sall is a senior research technician in the NYU Grossman School of Medicine Microscopy Lab. He has been a member of the lab for 8 years, specializing in 3D imaging modalities and analysis of both light and electron microscopy. He has his bachelor's in science in photographic sciences from the Rochester Institute of Technology. Thank you very much, all of you guys, for being here with me today. So to kick off, can any of you explain briefly what the protocol is about, how to use it, and why it is relevant?
Alice looks like she was going to explain.
I can start it. I think that this protocol is not very special compared with the previous published one, but particularly for this project, we put a lot of details into sample preparation and imaging acquisition. We have a long collaboration with the Gira's lab and did a serious electron microscopy work. And this is one of them. During this project, and Joey worked very closely with Noelle and try to narrow down each of the step, and especially the image acquisition. As you may know, that the serial block phase is the technology you can see the large volume in 3D easily, but the Z resolution is not easy to get accurate as the thing as we want. So with all those efforts put together, that's why we want to make it publish to let people know that when they when they are doing the similar experiment, they can follow our protocol and try to modify it according to their experiment, so that it will be benefit from our experience.
Yeah, and maybe if I can just add a little bit to what Alice said, for kind of new users of SBF-SEM, there's not a whole lot of detailed protocols out there of, if you're going to do something in cell culture especially. And so we kind of ran into that where we needed a good starting point, and finding that was a little bit challenging. And so we thought that since we had kind of systematically optimized several of the parameters, it could be useful to put it out there for other people.
As you were optimizing these parameters, what were the highs and the lows of the optimization? For example, is there anything that you tried and didn't work the way that you were hoping and didn't make it to the final version of the protocol?
I think particularly like Alice mentioned with the image acquisition, Joey and I spent a lot of time on the microscope kind of going through each of the parameters and kind of assessing which ones had different impacts on the readout in terms of what we acquired and the quality of the data. And I think that aspect was both fun because you can immediately see kind of some of the readouts, right, in image data quality, and kind of systematically going through it gave us a better appreciation of, all the intricacies of each of these parameters and how they affect what you're getting out. And as a new user, kind of to what Gira pointed out, it was very nice and useful to be able to get that hands-on experience. And so part of kind of putting out this protocol was also sort of being able to get people to be able to, simplify it for them a bit so they could jump right in, but also like see the kind of fun of being able to like play with all of these things as well. It is a lot of process, and so you spend many hours in a small room with a microscope, so that can be tiring, but I think it's equally fun to just be able to like play around with all these parameters and test them out. So Joey and I had quite a bit of fun just doing that.
Yeah, and to add to what Noelle's saying, in the end, this table we put together sort of distilled multiple years of experience with an SEM that highlights or correlates how an image would look when you adjust the specific parameters and why you would choose certain parameters. And so this whole, and you only really learn that after spending years and years with a microscope. And so the goal is to then maybe cut out some of that time factor for someone to pick up this technique or this method and more easily implement it.
So I want to add one point on there is not only imaging part, sample mounting is also another important step in here. And there's not really much detail written in the literature because this is a single layer of the cell, and you want the mounting to minimize the empty region so that the charge can get less during the imaging time. This is really challenge how to do it. So this part, I think we with Chris, our another technician's help, we developed the protocol which work well for not only this project and also for the future of the other projects. This is also very, very important. People may not notice a little detail like this can create a huge effect for the imaging. And this is another thing I want to add on.
At the beginning of the conversation, you mentioned that you wanted to lay out the ground for other people potentially to optimize the protocol for their own applications, given that the scarcity of detailed protocols in this field. Can you imagine what would be the potential applications and further optimizations of this protocol for other samples?
I think that this protocol will apply to any cultured mammalian cell line. When you cultured on the dish, this all can apply for using this protocol. And there's not much you need to tweak because we already write very detailed in terms of how to play the cell, how to get a set up mounting, and how to do the imaging. And in the meantime, if if in serial block phase, SEM field, if people want to get a nice Z resolution for the tissue and other organism, they can also use this protocol as a guideline, because that's how you should make notice. One is the height of the sample, another one minimize the raising, make the sample become more conductive, and the image parameter is also applicable to all the other applications.
I would like us to change gears a little bit. We have discussed the protocol, but I would like to know, in your case, what was the motivation to not only put together the protocol and post it in an open access repository like protocols.io, but have it peer-reviewed as a formal standalone paper in PLOS ONE?
That can go to Gira, because I would say that because this is Gira's project, we are the core facility. We have a long time collaboration, but this is an important question for them, and we just try to help as much as we can to establish the experimental result.
And so, if I understood correctly, your question was that why is it as a standalone protocol and not combined with the original publication?
No, my question, yeah, what motivated you to have this methodological paper peer-reviewed as a protocol paper?
I see. So there's, Noelle could also answer that, but there's a companion paper to this, which is the primary biological finding. And as we were kind of working through that paper and the methods of that paper, we realized, they were very extensive. And then we thought, well, maybe they'll actually be more useful if we kind of pull them out and put them separately, because we spent actually quite a lot of time optimizing this method. So I think sometimes it's a little bit just semantics of how you decide to put something out there, but I think it might have just got a little too buried if it was combined with the primary biological finding, which has been published in Nature Communications.
Yeah, exactly. To kind of Gira's point, as we were writing out the method section for the primary paper that's like of the biology that we were investigating using SPF-SEM, we realized that a lot of the nuance of the steps, because it is quite complicated and it's not very easy to parse, is lost when you kind of try to fit it in like a tiny paragraph. And it makes the technique seem really simple, which it is not. And so I think we wanted to be able, we kind of realized there was space not only to just create a more detailed protocol for people to see, but also like it would perhaps be useful for other new users, kind of like we were, to be able to kind of go to this and use it as a resource, should they want to.
And publishing it independently might also make it independently discoverable, right? Like, so people might not share your same biological interest,
Yes.
but they might be interested in the technique and find the technique independently of finding the Nature Communications paper.
Yes, absolutely.
And another point which you sort of allude to, Noelle, is that all the work, the put into the method potentially gets additional recognition, not only because of the biological question, but all the effort put into optimizing a method to make it work efficiently.
It is, because for any this kind of scientific project, one is focused on the project itself, and another one is for those, especially for us, like a core facility people, and how to help with different projects. There's people reading different journal, and for the technical people, they prefer to reading the technical journal because it's more easy to them. And also the detailed step-to-step material method will really help them to figure out how to resolve each question regarding different project. So whenever we have the chance, I think luckily Gira's lab is a highly collaborative and highly active lab, and they the postdoc and the PI in the school put a lot of attention to the project, and so that we can all work together to resolve the problem we're facing and try to get the project going on. And this is also something I want to mention. It's not every lab is willing to do that. You know, a lot of lab want to just based on their project going, they never care about the technical development of the details, but some other labs, everyone is really pay attention to the detail, try to get as much as possible, and also brought the scientific project to a different field so that, you know, they can use a different view to study their project. So we're very lucky to have Gira to be with us to working on this project. With also, you know, this kind of project usually it also help us to develop the technology. You know, sometimes the core facility, we're very busy doing daily work a lot, but when we have this kind of project, then we try to improve in terms of knowledge or everything and try to get the next level.
Yeah, and I think similarly sort of synergistic for us, because it's not always the case that the core facility is interested in doing something new with you necessarily. So, and I'm sure Joey will probably have a lot of complaints of how, you know, we really wanted to push the resolution and really, you know, have the data be like of the absolutely highest quality. And so I'm sure it was also frustrating, but we definitely appreciated it in the end.
No, it wasn't too frustrating actually. It was a worthy endeavor. Because these papers are exactly the sorts of things that when Alice and I go to volume electron microscopy talks or to any microscopy conferences, most people always say there's a dearth of these types of information available that really gets to the nitty-gritty of how things are done. And so this is exactly the kind of material that people are trying to advocate for more of, so that these techniques can be better disseminated and then further improved upon as more people are able to adopt them.
And how did the feedback that you received from the peer reviewers shape the final version of the protocol?
I think there were a few more like detailed comments and comparisons that they had requested that we kind of included. I don't think there were any kind of major shifts from what we originally had. We kind of did it at the similar time as the primary publication as well. And so I think by the time we had put it together, it was, I think, already pretty detailed and thorough.
Yeah, I think it really helps shape, like Gira said, kind of how easily the user might digest it. So it's suggestions of additional figures for like how to see more clearly the comparisons we made for like the image acquisition parameters and how that was affecting data quality. And so I think it helped with that and helped us sort of make more useful figures that could be helpful for a user.
And why did you decide to make the peer review history public together with the paper?
Sorry, you broke up a little for me. Can you say that again?
Yes. Why did you decide to make the reviewer comments, the peer review history, public together with the paper?
I think, I don't know about Alice can say for her group. For us, that's just the policy. I think we're pretty, we feel pretty favorable about everything being transparent. And so, you know, whether I'm the reviewer or the author, I'm happy for it to be transparent if anyone wants to see and make their own opinion and decision, right?
Yeah, totally agree. I think everything when we get into the technical level and transparency of the info, all those information is very important for everyone to get overview. You know, what's the problem and probably are facing, and if they can get more from the paper, that's what we want.
Yeah, and I think it also as more as reviewers, right? Like I think it's good for us to think of, you know, be able to write something that is that we are happy for it to be transparent too, and that's usually most helpful to the authors as well. So I think from both sides, that works well in my opinion.
And my last question, the answer to my last question might be a little bit speculative because the paper is relatively young. It was published less than 6 months ago. But how do you see, or what do you think will be the impact, the significance of having this methodological paper published for your careers, considering that you are, the four of you are at different stages?
Now, from the core facility point of view, it's always good for people to recognize us, and as they can continue follow up with our work, and we can also help when there's a more audience read the paper, they want to do the similar project, they can reach out to us. Not only the people from around and all over the world that they read the paper, they can contact with us and even send a sample to us, so then we can help them with the project. I think for the young generation, for example, like Joey, he really learned a lot during this whole processing, and he also present similar work will be present in the meeting, and so that more people can get benefit from this, and he also can use this opportunity as a better background for his own growth. And even though as a core facility member, we don't really count as how many paper published, because we do majority of the service, but this is really positive feedback when we get any review for anybody who did whatever thing breakthrough in the field, or more project related to some method development. This is really add advantage for his own growth and his own path.
Just I guess to add on to that, it does serve as a benchmark in a way, or where one's understanding of an imaging technique is at one point. And when you look back on a project and are able to see perhaps where one could then develop additionally from this paper. So this was sort of as an inspiration for further, I guess I could say, research or study that I wanted to do on exactly the idiosyncrasies of electron beams on sample performance. And so also this ties into the previous question of reviewer comments and how they really reinforce a need to have like a more deep understanding of the fundamentals of what's being said. And so these are the types of papers where you could address that exactly, where you can say why the certain decisions we made were made in this paper. And that serves as like a further foundation for later projects as you have that stronger understanding of the theories.
Yeah, I think for me, it was just overall a really positive collaborative experience to have in my career as a scientist, and working with like a co-facility and our team in the lab. So I think that was really nice. And it was also just such a great learning experience for just the literature. I had very little to no experience with EM and volume EM at that. And so I think it was just a great learning experience about the fundamentals, like Joey pointed out, as well as just the excitement of using microscopes. I work with light microscopy mostly, and so this was just exciting to be able to learn a different imaging modality and get into the weeds of the technique.
Well, I don't have any other questions. I don't know if there's anything that you would like to add that hasn't been discussed so far.
You know what? I think you covered
Yeah.
everything, but overall, this is a very fruitful and a grateful experience for us for the work, and we has been working with Gira's lab for at least 4 years on similar and and a different project, and this a positive experience. Really encourage us to do more in terms of technical development for the certain project, the equipment part, and this really will will add the protocol published, and we are be part of it, definitely will really give us more feedback from other people and make us more connection with all those people in the field, and even for the different field is we, as I said at the beginning, we don't have a lot of chance like this over my career as a core director. But from time to time, whenever we have this kind of opportunity, we are really grateful for the PI trying to spend time with us and try to improve the technology. This is based on their project, but this is a lot of effort they put in and try to figure out the things, try to resolve the problem. And so hopefully we can just get going in the future.
Yep, we can start a new project.
Yeah.
Well, thank you all very much. It was very illuminating, enlightening, the conversation, both technically and as well as your motivations to have the method peer reviewed.
Thank you.
Great, thank you.
Thank you. Bye.
I appreciate it.
Bye.